Okay, well, welcome back, everybody. I hope you've all had as good a break as we have. We've had a great, it's a nice refreshment. Now, we move into this session, as I say, which we're going to talk a little bit about the nuts and bolts of the library and how it's being used. As it's my enormous pleasure to introduce Holly, who's been doing a lot of the heavy lifting in the library in the last few years. So, Holly.
Thank you for that introduction, and welcome to our second morning session. As Martin said, I'm Dr. Holly Bratcher, and I am or have been responsible for the management of the data that is going into the MRF Genome Library since the project was created. And I first would like to begin by thanking the welcome singer, and in particular Craig Corton, who unfortunately wasn't able to join us today. Craig was integral in helping us to optimize the whole genome sequencing and also to establish the parameters for meningococcal genome assembly using the short read sequence technologies. So he really deserves a lot of credit for the initial work that was done as well. Apparently doesn't like my. Slight issue. There we go. We got it. So we have since that time moved the sequencing in-house to the Oxford Genomics Center. And one of the things that has allowed us to do is to have a quicker turnaround time for the samples that are submitted and getting the genomes into the database along with the isolate information. So we've heard quite a lot about this already this morning, but just really quickly to highlight, the goal of the genome library itself was to provide and make available a coherent set of high quality meningococcal genome sequences that could be annotated not only for research purposes, but also as a source for vaccine development, the benefit of public health measures, and consequently, the other thing is that it has allowed us to do is set up a series of validation methods that can be used and shared across the community. In order to achieve these goals from the beginning of the first genome sequences that we have done, several of the key processing nodes were not only tracked, but then the data was analyzed in conjunction with these different steps. So everything from the extraction protocol to the library creation and the sequencing technologies straight through to the curation protocols that we use in-house to actually annotate the genomes. I've been tracking since the beginning of this project, so we can see how this data is changing, the quality is improving, or where we're having issues, and whether or not a new method that's been introduced, we're able to detect these changes and what impact they have. Because as most of us who work on the genome sequences know, this is quite a difficult genome to work with because of the recombination and also the actual architecture of the genome with lots of repeat sequences in it. So we need to be able to understand when we see changes or why things maybe don't assemble properly, what is causing this. Overall, over the 10 years that we've been doing this, we have continued to see an increase in the contig length because of and a decrease in the contig count. And this is really important because it means that these repeat regions within the genomes are starting to be assembled more and more. We're being able to recover more of the genes that are in the genomes themselves. And this has been down to a couple of different things, not only using different extraction techniques, but primarily the technologies have been advancing with the sequencers themselves. We are constantly looking at and keeping up to date with what technologies are available and being used. I work quite closely with the genome centers, and we have a set of data that we do periodically test to see how the new technologies will handle the complexities of the meningococcal genome. The second portion of that is also the sequencing chemistries. And so for short read sequencing, the chemistries are advancing. When we first started doing this, a short read sequence was about 57 base pairs long. If you can try to imagine to assemble the 2-point KB that we needed to with 57 base pair pieces, this took an awful long time. And a lot of sequences were not fully assembled because of this. We have now moved on and we are approaching 300 base pair reads, which is quite substantial for the meningococcal genome. And in most cases, is it's starting to bridge a lot of the repeat regions, although we still have quite a long way to go on that. So one of the things I'd like to mention about that is this optimization that has come out of being able to track all these things has allowed us to share this information and help other groups to optimize the protocols and the pipelines that they're putting together as they're moving forward in meningococcal genome sequencing, so that when the data is put into the larger Neisseria database, we can use it to compare to the MRF genome, in particular other countries' data, or when we're looking at perhaps outbreak situations and things like that. We have an easier time comparing these things together. So it's quite an advantage to have this vast amount of data. Quickly, I also wanted to say that the other thing that we do is we do closely monitor all the loci that are in the database that we use for the annotation process. And the reason we do this, like I said, is because not all of these loci assemble very well. And we want to make sure that the core genome that we develop is representative of all of the types of Neisseria that we see, whether it's in an IMD case, a carriage case, whether it is something from the UK or from somewhere else. We want to make sure that we are capturing that information in the best way possible. So we do monitor these. Now, the MRF Genome Library has played a critical role in the development of a lot of the schemes we do for typing. And initially, when we set up the core genome library, the core genome MLST, you can see right here, it had 1,600 loci in it. And we had a threshold on that of 95%, basically because of the draft nature of the genomes, but also because we wanted to have a little bit of flexibility in understanding what the population biology looked like. We didn't know if all of the genes we were seeing in the relatively small data set we had were core or not. So we needed to have that flexibility to see how it changed over time, because genomes and the population biology itself is quite variable. And we needed to track that. So continuing on to present day, and the genome library is still a critical component of looking at the development of the schemes, and in particular the core genome. And very shortly, I hope to be able to release version 2 of the core genome MLST. And I'm expecting this to have around 1,400, possibly 1,500 loci in it. We are testing this against a couple of different systems, including tightening up that threshold for what is included and what isn't. And what we're looking at is how the lineage affects the number of genes that are in this. So we want to be able to capture lineage splits and lineage information, but at the same time be inclusive of all of the genomes and the characteristics that we have. So moving on, I think hopefully some of this presentation that I'm going to give, as well as the rest of the people in our hour, will answer some of the questions that were brought up in the last session about the structuring and how this is based. So as everyone knows, the MRF Genome Library is embedded within the PubMLST database, and we are constantly curating, data cleaning, and making sure that the information in there is of the highest quality possible. This is done through an iterative process within the system itself of PubMLST and developing the systems, but it is also done through a process where we are constantly outreaching to not only our curators, but the submitters to the genome library, and then getting their feedback as to what's important, what's working, what's not working. So in addition to the curation and the upkeep of the data that we're managing, we do data mining both within and external to keep this information in a manner that is of high quality. So what does the structure of the data look like? So we use a gene-by-gene annotation process that allows us to look at everything from the fragments of typing for something like MLST, or the mutations that occur in antibiotic resistance genes, all the way up to the capsule typing operons, which is quite a large chunk of the meningococcal genome. But we can also look at metabolic processes such as the iron acquisition. And all of these together help form the structure that we know as the population that is the meningococcal, but also is the structure of the genomes that we're looking at. Excuse me. So those are the basic things that drive it. So one of the things that we had to do as we were setting up the database and setting up the actual data capture of this was to make sure that we're including all of the information that's relevant. So currently, the genome library has over 5,000 meningococcal genomes that have been recovered from IMD cases here in the UK. And it also serves as a sentinel surveillance, as Jane mentioned earlier, for a lot of the disease that is occurring in the UK. And a lot of the surveillance things that are looked at are everything from the capsule type to the clonal complex, but we're also starting to capture antibody information, vaccine coverage information within that as well. We had a question earlier about capturing the phenotype. This is also something that we're working on bringing into this data structuring. And we're hoping that over time, working with PHE, the MRF Genome Library, as well as collaborators and curators, that we can bring more of this into the system. And we'll be hearing a little bit more about that later on in this hour's talk. So the Genome Library currently holds enough data, both specific and general, that we can look back over time, and it provides a historic perspective, but it also provides enough characterization that in the low levels that we're seeing in the past 2 years of disease, we still have enough information to judge the characteristics and annotate and look at the structure of the population currently, even with the low recovery or infection that we're currently looking at. And through all of this, what we can do is we can draw conclusions about the lineage development, virulence factors, and prevalence, and that we're going to be looking at later. So you've seen this all before as well. One of the things that has been, I think, most striking to what we've been able to do is the capturing of the change that we've seen over the years, and in particular, being able to pick up the change the increase of the meningococcal type, capsule type W prevalence that began, and then the expansion in particular of the, oops, sorry, I've lost my cursor, there we go, of the ST11 complex here, and we can see how that has actually changed over time from a phylogenetic perspective. And so we can ask a lot of questions about not only when and why that's happened, but how that's happened by having the meningococcal genome library. We can dig into this more specifically. But I think one of the things we have to keep in mind with the data structure is it is more than just the individual phenotypes that we're seeing. It is more than just the characterization of individual genes that we use for tracking and surveillance. And the links that we need to start looking into now, and that we're hoping to start to incorporate into the database along with the genomes, is included in the isolate records. And so each genome, we request the submission of an isolate record that includes provenance, epidemiology, and the demographics of the isolates. Now, we don't require all of this information, but these 9 points of data is something that we would highly like to see increased within the database, and suggest that the submitters include. And what this allows us to do is stratify the data in such a way that it brings those links that are more about the biology of the organism into matching with the phenotype. So it's this phenotype genotype link that we're looking to provide with some of this thing. So just as a quick example, and keeping in mind that we do have some data gaps here, the example I'm showing you here is age range, which we can then link to some of the genetic information that we have within the library itself. So if we take a look at the region, and we use a NUTS classification, which is a statistical demographic of the UK that is used, along with the epidemiological year, and like I said, the age range, and we're going to be looking at, in particular here, the capsule type. This is one of the visualizations that we're developing, so we can start to bring together all of this information in a way that is more connected, where you can actually look at data instead of in a graph or a table or a chart. You can see the links in the data. Now, I realize this one looks a bit messy at the moment, and if you can see here up at the top, this is a snapshot of the Sankey dashboard. I have here listed all of the epidemiological years for the meningococcal library, all of the regions that we have in, and all of the age groups. So that is everything that we know at the moment. So for example, if you wanted to take a look at a specific region, you click on the region bar here, and the example I've got here is for Yorkshire and the Humber. And you can, if you're a clinician or a public health authority or agency that is in that region, you can look specifically at the data that is in your region from this, and we can see over the years what has been the most prevalent as far as capsule group, and this is also providing us, in this particular example, the clonal complexes. So this can give the public health a lot of information about what is, where the context of their isolates could occur. And in particular, if you were a clinician that perhaps, or a GP that has a case that you're trying to look at and you want to understand better where that fits within the larger picture of your region, you can actually even look at this data even more specifically as far as if, for example, your patient was a child under 1, or also I guess would be considered an infant. And you can start to see how this information can link back to providing a public health side with some information that can be used more constructively than just looking at charts and graphs and totals and summaries of data information. So by linking all of this information together, we're able to then see how it's connected. We can start to overlay this with phenotype information. And as I showed earlier in one of the slides, the more of this information that we can provide in a manner that's helpful, the better. And Scotland, our colleagues from Scotland and Keith later on will be sharing with you some more information about how this structure is developing for these types of things to move forward. And I think finally what I'd like to say is that I found it really hard to try and compress everything into 15 minutes for the structure and the validation. Suffice it to say, I've been doing this for 10 years, and it does feel like it's taken 10 years to get to the position we are at now. There is a lot of data there, and a great thanks goes out to all of our curators and people who have contributed to doing this. And as you can see here, it is a substantial part of the Neisseria database that PubMLST does host, and we'd like to see this to continue to grow, not only in and of itself, but then as a part of the larger global approach to looking at this and being able to use this data for the betterment of public health and or vaccine policy. So this is helping us to start to unravel these questions about linking phenotype to genotype, understanding how that population develops, and then how we can use that potentially in something like predictive models, but also on the clinical side so that they're able to have some real-time information so that they see the benefit of the genome sequencing and the MRF genome library within that. So as I said, these foundations of the structure and the validation that we have been working on over the past 10 years is now allowing us to move forward with some more practical tools for the wider community as well as public health, and hopefully also benefiting some of the people that participate in the MRF from the participant side, from the patient side. Thank you.